Hi everyone, welcome to another video from Shomu's Biology. In this video lecture, we are going to talk about the protein translocation inside mitochondria. This is the third lecture of our protein trafficking and sorting series. So I want you to watch my other lectures for a better understanding of this particular lecture and to the total understanding of protein trafficking in the cell. But in this particular lecture, we are going to talk about the translocation of protein inside the mitochondria. Means basically how a protein after the synthesis in the cytosol is transported inside the mitochondrial matrix. That's what we need to understand in this particular video. So here, what I can say is very simple, very clear. And this understanding is the protein is produced in the cytosol. So so let me just erase all this color from here once. Okay. Okay, I'll take a different color. Okay, so what I'll explain here is the journey. The journey is from the cytosol. Okay, from cytosol to the mitochondria. Cytosol to the mitochondria. That is the directionality, that is the journey of the protein. Produced in the cytosol, and as we know that the proteins which are destined to be delivered inside organelles are not going to take the path through the Golgi apparatus and all. So they will only be produced inside the cytosol, directly transported inside the target organelle. They need to have a specific signal. They need to have a specific signal to them. Sorry for this. There's an issue with the writing device here. That's why this issue is going on. So the signal, there's a signal sequence to it. The signal sequence is a mitochondrial localization signal. If it is destined towards nucleus, there is a nuclear localization signal. This sort of signals means basically some stretch of amino acid sequence, specific amino acid sequence required for the protein to be targeted towards mitochondria. Okay. So in this case, once that signal is present in a protein, the protein is destined to be delivered inside the mitochondria. But once it is ready to be delivered inside the mitochondria, then there are a set of events involved in the process of mitochondrial translocation. Basically, when I say translocation, means basically the proteins produced in the cytosol will be transported inside the mitochondria. And we know in the mitochondria there are different locations. There are outer membrane of mitochondria. So we can say the mitochondrial outer membrane. There are mitochondrial inner membrane. Mitochondrial inner membrane, or there is a mitochondrial matrix. So the protein can be delivered to any of this place, either inner membrane or outer membrane of mitochondria or inside the mitochondrial matrix. In all these cases, the delivery is done through specific channel type proteins in the cell membrane, in the membrane of mitochondria. So outer membrane of mitochondria has some sort of these channel proteins. Inner membrane of mitochondria also carry some of these channel proteins. They have different names, generally termed as TIM and TOM. I must write it here. TOM, Tom. This is the translocase to the outer membrane. So the translocon, that is a channel protein to the outer membrane. And there is TIM, translocase of the inner membrane. So TOM is produced and packaged in the outer membrane of mitochondria. TIM is produced and packaged to the inner membrane of mitochondria. And there are also other complexes like OXA, SAM. All these complexes are there. They have their own role. We are going to discuss that later. And also for this process of protein transport inside mitochondria, remember one thing, the protein must not be folded in the cytosol. Because if the protein is folded, it will be bigger. It cannot be translocated through the TOM or TIM complex. So for the protein's delivery to the mitochondria, we need the proteins to be in the polypeptide state, nascent state, that is in the linear polypeptide state. And the folding will be done after it is transferred inside the mitochondria, right? So for a proper folding, for a guided folding, because if the protein is present as nascent peptide, it can misfold. So to prevent misfolding, we also have heat shock protein, also known as chaperones. Basically heat shock 70 and each of protein 90 involved in the process of mitochondrial protein delivery. Got it? Now we are going to share you a list of important parameters for the delivery of proteins inside mitochondria, translocation of protein inside mitochondria. Okay, what are they? Let's move on. Here I can discuss. So I'll also change the color. I'll take blue color here. We'll start discussion. The protein synthesis part means the synthesis is done in where? Cytosol. No issue with that. Cytosolic protein synthesis. And then the protein will be guided by chaperones. Chaperone guiding, HSP70. There are chaperones present in the cytosol, known as cytosolic chaperones. And there are chaperones present inside mitochondrial matrix, known as mitochondria. Here cytosolic chaperones will guide that protein towards the mitochondria, because the mitochondrial delivery protein has a localization signal to it. What are the import receptors? The import receptors are TOM complex, like TOM22, TOM20 and TOM40. These are the complexes known as import receptors, which can interact to the destined protein and can bring it inside. So that is the import receptors. Then translocation through TOM complex. Obviously the translocation can be done. TOM22 is involved in the process of delivery and translocation. Translocation through TIM complex is the next step. TIM complex is the complex of inner membrane. Translocase inner membrane TIM. Translocase outer membrane TOM. So TIM23 involved. TIM17 is involved in this process. 1723 they are involved in the transport. Then folding and modification. Right after the transport via TIM, there will be folding and modification inside the mitochondrial matrix. So the folding is done. Again, HSP 70 is necessary and required, but these are mitochondrial HSP 70. So the folding and modification will be done in a mitochondrial matrix by HSP 70. Right after the folding and modification, sorting and subcompartmentalization is done. Means we know in a mitochondria, if you look at the mitochondrial structure, in very simple words, if I draw the structure here, it looks something like this. There are two membranes, so I'll draw both the membranes. This is inner membranes with Christie. I'll draw like this. So this is outer membrane, this is inner membrane, this is intermembrane space, and this is matrix. So all these different components are there, compartments are there in a mitochondria. So the proteins transferred inside mitochondria are destined to be delivered in different compartments. It can be delivered into the outer membrane, or embedded to the inner membrane, or delivered to the matrix, or it can be transported inside the intermembrane space. So this further compartmentalization is done. Right after the compartmentalization, there will be assembly into complexes. Now this step is not common to every single kind of protein in the mitochondria, because there are some proteins inside mitochondria requires further assembly. They are transported in fragments and then they are assembled inside the mitochondrial matrix. And the example of these assembly complexes are mitochondrial ribosome. Mitochondrial ribosome is involved there. Similarly, what else is involved here along with mitochondrial ribosome? Another protein involved along with mitochondrial ribosome, that is the ATP synthase. Okay. Both of these enzymes, ribosome and that enzyme both require further assembly that is done here. After that comes to maintenance and degradation. So the maintenance will be done. And here what is done is maintenance and degradation. And why it is important to have a proper maintenance and degradation is that without proper maintenance and degradation, we cannot support the presence of the organs, of those proteins in the mitochondria. Because inside the mitochondria, the proteins, once not folded properly, they are misfolded, they will be targeted for destruction. And that pathway is known as unfolded protein response pathway, UPR, unfolded protein response pathway. This unfolded protein response pathway ultimately leads to the destruction of unfolded proteins and thus regulating the proteins final fate. So it's like a quality control for the mitochondrial proteins at the end. Okay, so these are the overall steps. So these are the overall steps for the process of mitochondrial protein translocation in sequence, in that order. Starts with protein synthesis in cytosol, then import receptor binding, translocation through TOM complex, outer membrane complex, like TOM22, translocation through TIM complex, inner membrane complex, like TIM23, folding and modification in the mitochondrial matrix by HSP70, then sorting to subcompartments like delivering it to the outer membrane, inner membrane, intermembrane space or matrix of the mitochondria, then assembly into complexes, few cases ribosome ATP synthase, and then finally maintenance and degradation. If the protein is good and fine, it should be kept there. If the protein is not good, misfolded, then UPR, unfolded protein response systems, will tag it and degrade it by mitochondrial protease. Many students ask, like, what kind of degradation is there? Ubiquity? No. This is degraded by mitochondrial protease enzymes that are present inside the mitochondria. Now, one big question still left. I'll move to the next slide to discuss about how the proteins are destined to the different sections of a mitochondria, different compartments of a mitochondria. But before going in there, I must say that one question is always there. This process of mitochondrial protein delivery, do they really require, do they really need ATP? The answer to that is yes, they require ATP, because the proteins that help the translocation of protein inside mitochondria, those proteins production and maintenance itself requires ATP. For example, TOM complex, assembly, maintenance requires ATP. TIM complex, maintenance and assembly requires ATP. HSP, which are heat shock protein, also known as chaperones, require ATP for their normal functioning. So we need ATP for the normal functioning of all this process regarding the mitochondrial protein translocation. I believe you have a clear understanding of mitochondrial protein translocation now. If you understood this pathway clearly, it's fine. We'll move to the next slide showing you the involvement of different proteins like OXA, SAM, TIM, TOM, all these complexes and how exactly they're involved in terms of delivering a protein in their destined location inside mitochondria. तो हिंदी में सिंपली एक ही चीज आपको बताना है, तीन इंपॉर्टेंट फैक्ट्स. एक तो है कि द प्रोटीन्स जो प्रोड्यूस होता है साइटोसॉल में वो डायरेक्टली डिलीवर होगा साइटोसॉल से माइटोकॉन्ड्रिया में, स्पेसिफिक माइटोकॉन्ड्रियल लोकलाइजेशन सिग्नल है. तो दूसरी चीज, ये जो डिलीवरी होगा प्रोटीन का, ये माइटोकॉन्ड्रियल मेम्ब्रेन में जो प्रोटीन्स मौजूद होता है टॉम टीम, इसके जरिए होगा. और इसमें एटीपी यूटिलाइजेशन होता है इनडायरेक्टली और डायरेक्टली इन बोथ दिस केसेस, जहां पे टीम टॉम को मेंटेनेंस में लगता है, एचएसपी की मेंटेनेंस में लगता है. और तीसरी चीज, जो फोल्डिंग ऑफ प्रोटीन होता है वो अंदर जाने के बाद ही होता है और उसको डिलीवर किया जाता है सब कंपार्टमेंट में. कैसे सब कंपार्टमेंट में डिलीवर करना है वो चीज अब देख लेते हैं. ऑलराइट, सो नाउ वी केम टू दिस स्लाइड एंड आई वांट टू डिस्क्राइब द प्रोसेस ऑफ protein delivery in the mitochondria, particularly the localized different compartments of mitochondria. For that, we involve with several different proteins. We know that this is where the ribosome, this is the protein that is synthesized. The nascent mRNA, from there the nascent protein is produced. Nascent protein means the linear protein that is produced, the precursor protein that is produced. Then this precursor protein will be delivered inside the mitochondria. So either the delivery location is mitochondrial matrix or to the inner membrane or to the outer membrane. Any of this is the delivery location. And you can clearly see that the nascent protein can take the journey via TOM, because TOM is the response protein which is going to interact to the signal, to the mitochondrial localization signal of the nascent polypeptide. So obviously the first set of protein that the precursor protein should be in contact is the TOM. So from TOM, this protein can take the journey via TIM here and into the matrix. So if you need to deliver to the matrix, TOM is obviously there for all these three, but if you deliver to the matrix we need TIM. So TIM is needed along with TOM to deliver the protein into the matrix. Now if you want to deliver protein into the inner membrane of mitochondria, like this one or like this one, what else we'll use? We'll use TIM22. So in case of direct transfer of the protein from the cytosol into the matrix of the mitochondria, TOM is used, then TIM23 is used. So you write TIM23. TIM23 is used. While if we deliver the protein to the inner membrane, we can either go TIM22 or OXA complex. So here in the inner membrane, same TIM, but TIM22 is involved in this case. TIM22 or OXA complex is used here. Okay, either via OXA, but if the OXA is involved, then TIM23 or OXA can do that. But remember, this precursor protein which is transported into the matrix directly requires TOM and TIM23. And if TOM and TIM22 is involved, then the protein can be embedded to the inner membrane of mitochondria. No issue, fine. But there is a big issue with OXA, because OXA cannot directly take that precursor protein from cytosol and embed it to the inner membrane. Only TOM and TIM22 can do that. TOM and TIM23 can drag it to the matrix. But OXA's role is when the protein is present to the matrix, then that protein can be embedded to the inner membrane by OXA. So OXA's role is not to transfer the protein from cytosol to the inner membrane, but from the mitochondrial matrix to the inner membrane. Remember that. Keep that in your mind. And lastly, if you want to deliver a protein to the outer membrane, then use SAM complex. SAM complex. And then you embed it to the outer membrane. Okay. So this is very important to understand. So TOM complex is always used. I must write it in here. So next cases. So TOM TIM23. I'll state this. Take it here. So I have TOM and TIM23. This is one approach. I have TOM and TIM22. We have another approach, and we have directly the other hand SAM complex, TOM and SAM complex. We have another approach, and lastly we have TOM TIM23 and OXA. We have a different approach. These are all the approaches from bringing the protein from cytosol to the different compartment of mitochondria, right? And the only case where we use OXA, only OXA, that will be involving a protein's movement from matrix to inner mitochondrial membrane. Remember that. I'll take a different color. To write down the important steps now. TOM and TIM23 brings a protein from cytosol to matrix. TOM and TIM22 brings a protein from cytosol to inner mitochondrial membrane. TOM and SAM brings a protein from cytosol to where? Outer mitochondrial membrane. And TOM and TIM both, remember, both they bring a protein to the matrix from cytosol. Then OXA can take the protein from matrix and put it to the inner mitochondrial membrane. And directly only OXA will transfer the protein from mitochondrial matrix to the inner mitochondrial membrane. That's almost very simple and basic way to explain the role of different proteins, mitochondrial proteins, in the process of mitochondrial protein translocation. I believe we have a clear understanding of mitochondrial protein translocation with this particular lecture. If you like this video, please hit the like button, share this video with your friends, and subscribe to get more videos like that in future. Thank you. Bye.